Hello everyone, and thank you so much for joining my talk today. Before I start diving into these super interesting topics, total RNA sequencing and metagenomics, I want to address a more basic topic first, which is ecological assessments. As we all know, ecological assessments are used to classify the status of ecosystems. And in the EU, for freshwaters, this is controlled by the EU Water Framework Directive. This directive has the goal to establish a good status of all EU water bodies until the year 2027. And now the big question that was asked in the past is, what is this good status? How do we define this? And this definition has been changed over time based on our results of research we did, and the current definition is that ecological quality is defined by the response of the biota rather than by changes in physical or chemical variables. So we see the biota is the key here, and many people think in this context of fish, of plants, of diatoms, but actually, if we look at the whole biodiversity on the planet, there's much more than just plants or animals. We have a ton of bacterial species, we have a ton of archaeal species, and a ton of single-cellular eukaryotes. These are all classified as microbes. And recently we realized microbes actually make really good indicators as well, and we should include them into our ecological assessments. Now for microbes especially, but also for everything that we do here on the conference, we apply metabarcoding. So I'm not gonna invent the wheel new here by talking about what is metabarcoding. I assume you all know what it is, and if you know what it is, then you also know that metabarcoding is biased. There's primer bias and there's PCR bias. And today in this talk, I'm going to talk about an alternative solution for taxonomic profiling of the biodiversity in samples. This alternative solution is shotgun sequencing. That means we use usually a machine to fragment DNA or RNA by sending waves into our samples that will fragment the strands. If we fragment DNA, we call it metagenomics. If we fragment total RNA, we call it total RNA sequencing. And I would like to point out that total RNA means RNA that has not undergone ribosomal RNA depletion. If we apply those approaches, there's no metabarcoding needed. That means there's no primer and no PCR bias involved. And at the same time, we are able to investigate the entire biodiversity and not only groups that our primers are targeting. Let's briefly compare those two approaches, metagenomics versus total RNA sequencing. And the key element in this comparison is SSU ribosomal RNA read abundance. If we look at the tree of life again, we have determined specific genes, so-called barcodes, for the different groups we are interested in. For Opisthokonta, so animals and fungi, CO1 works best for animals, ITS for fungi. I know there's exceptions to the rule, but this is just a general proof of concept. For plants, RBCL has established as the best barcode. But now let's quickly, politely ignore those two groups and look at everything else, because for everything else, SSU ribosomal RNA, so small subunit ribosomal RNA, has been established as the best working barcode. Exceptions excluded here. So what does that mean if we apply metagenomics to a sample? Imagine this circle here is the entire read pool that you get from a sample, all the reads. What I'm going to do now is I'm going to show which proportion of this would be SSU ribosomal RNA. And as you can see, not much, to be honest, only 0.05 to 1.4%. And that is because in metagenomics, we look at all genes in a sample, not only at one specific gene. So this gene we are interested in for microbial taxonomic profiling just gets a tiny portion of the reads. And if we consider that this is used to taxonomically identify the gigantic portion of the biodiversity, that is not much. Now in comparison, if we apply total RNA sequencing, Same situation. This is the entire read pool. This is what would be made up by SSU ribosomal RNA, 37% to 71%. Much more. And the reason for that is because RNA consists of 80 to 90% of ribosomal RNA. So a big portion of it will be SSU ribosomal RNA. Now, it seems that if we want to identify the majority of the tree of life, this seems to be the preferable approach. But actually, that hasn't been tested yet. So in this talk, I'm going to address the question, which of those two performs better for taxonomic profiling? To tackle this question, I came up with the following study design. I bought a microbial mock community consisting of 8 bacterial and 2 eukaryotic species, and I sampled a display tank that contained a diversity of organisms. For each of those sample types, I created a water sample and had 3 replicates each, which I then filtered on 0.2 micrometer filters. I extracted the DNA and the RNA in parallel and shotgun sequenced them on an Illumina MySeq. Then I processed the data, and this step was actually so intense and time consuming it deserves its own slide. And lastly, I determined the accuracy and precision for each workflow that I applied. As promised, I'm talking about the data processing on its own slide, and for the data processing I applied 6 steps. Step 1 was trimming and quality filtering, where I used 4 different Phred scores. Step 2 was ribosomal RNA sorting, where I sorted my reads into ribosomal and non-ribosomal RNA. Step 3 was the assembly, where I used 4 DNA-optimized assemblers and 4 RNA-optimized assemblers. Step 4 was mapping, where I mapped my reads back to my assembled contigs. Step 5 was the reference database. I used the Wild West and CBINT and conserved silver. And step 6 is the taxonomic classification, for which I used 3 different approaches. And now what I did was testing the combination of all possible tools on all my samples. So each of the combination of tools I refer to as a pipeline, and these sum up to an amount of 1,532 pipelines that I ran on DNA and on RNA samples of mock community and fish tank. Now let's talk about my preliminary results. So far, I looked at the mock community total RNA sequencing data based on the species level identification. And I plotted every pipeline into the same graph, which resulted in this graph, and each dot represents a pipeline. For each pipeline, I estimated the accuracy based on the expected community composition versus the observed community composition for the pipeline, and the precision across my 3 replicates. I normalized the axes from 0 to 1 based on the lowest and highest values I found for each of those parameters. And now keep in mind, when looking at this graph, the pipeline performance increases in this direction on the x and y axis, so that the higher top right a pipeline is, the better it performed. Now, obviously we can't see much in this graph here, it's just a bunch of dots. So what I did, I colored them based on the tools I used in each of the steps. First, I colored the pipelines based on the ribosomal RNA filter I applied. And there's a clear pattern visible. If you can't see it right now, then you should latest see it now. If I color them based on sort.me RNA, we see that pipelines that involve sort.me RNA on average perform better in terms of the accuracy than all the other pipelines, because this cloud is further on the right side of the x-axis. If we color the pipelines based on the applied assembler, we see that pipelines that involve IDP8 trend for assembly perform on average much better actually than all the other pipelines. And if we color the pipelines based on the classification tool used, we see that pipelines that involve Kraken2 outperform, on average, the other pipelines that involve BLAST, which was pretty surprising to me, to be honest. What was also pretty surprising to me was that it didn't make a difference if I used the NCB INT database or the silver database. There was no clear pattern in the distribution of pipelines, which I didn't expect, to be honest. So what preliminary conclusions can we draw from this? So far, we can only draw conclusions for total RNA sequencing. Conclusion number 1, there are big differences in accuracy and precision of pipelines, clearly visible in our graphs, and some tools are on average more accurate than others. These are for ribosomal RNA filtering, sort-me RNA, clearly, for assembly IDBA-TRAN, and for classification Kraken2. And last, I would like to talk about what's next in my project. So far, I talked a lot about, or actually only about total RNA sequencing, although my talk is called Metagenomics in comparison to total RNA sequencing. So what is going to happen next? If we again have a look at the graph here that I showed you before, these results here are only based on total RNA sequencing. But what I will do in the future is that I will add the metagenomics data. So eventually this graph will have twice as many dots, twice as many pipelines. And that way I am able to see if the pipelines based on DNA or based on RNA perform better in comparison. Also, I haven't had the chance to have a deeper look at those top right pipelines, which are supposed to be the best pipelines, and this is what I'm going to do in the future as well as evaluating my analysis based on other taxonomic ranks, not only the species rank. And eventually I will compare the results I get with my mock community to results I get when sampling the display tank. And with this I come to the end of my talk, and I would like to thank my amazing lab, I would like to thank my amazing collaborators, and some other amazing people that gave me great advice for this project, as well as my funders. And if you have any questions, don't hesitate to ask me. Thank you so much.